So you've all seen the cortical folding maybe, and you know that the cortex fold in mammalian brains, and we're not the most folded species. And you've probably also all seen the textbook examples of the names of classical nomenclature of the cortical folds and the gyruses. But in reality, the variability, there's a huge variability that exists from one individual to another. If you look at, for example, even the central sulcus, there's already some shape differences. And then if you look at the green one, for example, the superior frontal sulcus, then the variability becomes very big. So normally, what we see is that, for example, we see a textbook example of the central sulcus is an average of all the other individual folds, and then if you observe the individual folds, some of them are more flat, the other are more curvy, and so there's a huge variability that exists. So if we go to the area of the pre-central sulcus, including and the central sulcus, the pre-central gyrus, as you can see at the bottom, the variability becomes really huge. And so there's a collaboration that was done between the group in Neurospin and the Yiddish brain group, and a great work tried to map the brains of the Yiddish brain onto the sulcal folding. And despite the variability, there are some results that shows that, for example, if you look at the area 44 and the brain at the left, and it's right next to the precentral, inferior precentral sulcus, but then at the left, at the right, for example, sometimes there are some variabilities of the cortical folding, and it's as if the inferior precentral gyrus. So here you can see the variability, and probably it's actually not anymore right next to the precentral sulcus, but at the ramus, diagonal ramus of the Sylvan fissure. And so the tool that we used is developed in Neurospin. It's called BrainVisa software platform. There was a talk by Denis Rivière at the workshop to introduce the software morphologists and anatomists, and used for brain segmentation, sulcal analysis, automatic detection, labeling, and visualization of cortical folding. So this is the tool that we used, and we obtained a mesh, and then we peeled the cortical mantle. We have a negative cast, and after that, there's the automatic labeling that will give us the final names of the sulci. Since it's last talk, everybody's hungry, and it's in Italy, so it's the idea of really mixed up in terms of cortical folding patterns of all these. For example, the example of the central sulcus. And so the idea is try to understand if there is a variance, a difference that we can understand. So the simple idea is to make a similarity measure. And so here, for example, anyways, so you can make a similarity matrix. For example, when the coding shapes are very similar, so the distance will be small, and when very different, distance will be bigger. And the idea is simply to do a dimension reduction so that we project this data from a higher dimension to a lower dimension, so that we can have some meaningful interpretation of the variability. So isomap is the algorithm that we used. It's a simple dimension reduction algorithm that finds the neighborhood of similarity, and then we simply go down, for example, from 2D to 1D, so that we can plot in one line of the variability. So you probably all know there's this knob that's called the hand knob that's mapped. You see a little star next to the knob. That's the characteristic shape of the hand activation on the central sulcus. And before we organize it, and so the first is the random order. You can see of the central sulcus. Again, it's very variable. And then after that, when it's ordered according to the similarity, and if you observe the hand knob from left to right, you can see that gradually goes up. And then on the extreme side, there's a little knob that appears kind of at the bottom. So if we have more subject and we plot it all together, you can see still this tendency from left to right of this hand knob that gradually goes up and the second knob that appears. And to be able to really look at the shape, we do simply average shape, which at position 1 or 2, we make a weighted average. And we make an image that's averaged, and then we make a mesh out of it, so that we can observe the shape at a particular place at the axis. So again, this side is more a single knob. We call it a single knob. There's only one knob. And the other side is a double knob organization. So if we plot it onto one axis, you can observe that at one side there's a more single knob organization, and the other side there are two knobs. We call it essential variability. It's like the idea of the PCA, and we have one major variability trait, which is the moving knob. And so we try to make a link of this kind of study of the cortical folding with the functional activation. There's this study that has hand click and silent reading of the function. So the hand click is the red one, and you can see that it follows the hand knob. It goes up, and so it confirms that there's some truth in the variability, and it follows the activation. But then when you look at the silent reading activation, it looks like it's going away from around the second knob. But when we look actually at the precentral gyrus, if we plot also the precentral sulcus, you can see that the activation is actually moving into the interruption. So we used here the HCP data set of 882 subjects, and to do the same kind of study, just with more subjects. And as you can see, that then it's the idea that we can have different summaries, like the PC idea to summarize the variability traits and plot the average shape. So the first line again is the second knob, single knob trait. And then if you look at the second line, you can see, we also see at one side is more curvy, the other side is more flat. So, and the third again, so we can actually look at the real shape to see why and how they differ. So again, from the HCP data set, and there's the hand activation. We plotted into this axis of the shape variability. And you can see from, again, the activation follows the knob from one side to the other. And also the position, that's the position, also the magnitude is bigger when there is a bigger hand knob. And going to the more complex area, the precentral gyrus, again, if we look at the variability traits, and the first one, if you look at the inferior precentral sulcus, so there is from an I at one side to kind of a T. From I to a T configuration. And the second one is, it looks like the precentral sulcus is more interrupted. And the third dimension, you can see it's like a nail, kind of a little nail form to a more I configuration. So Finally, we look at the activation of the story versus the baseline of the HCP dataset. And as you can see that there are two places of activation and one at around 44 area, and then the other one is higher, and it looks like it's going to cross. It's crossing the sulcus. So looking more closely at the area 44, and again, as you can see that from the eye here to the T shape, and it's getting bigger in activation. It's closer, right stopped at the T activation. And then if we look at area 55, this is plotted against the third dimension from the little nail kind of configuration I was talking about, and to the eye configuration. And if we look at this dimension, we can see that actually the activation is going into the interruption, and it's going from more horizontal to a more vertical shape. So finally, if we look at a real cell size at this average shape axis, and you can see that actually the inferior precentral sulcus here is going from a more horizontal location here, gradually going more vertical until it really goes more vertical. And here are the zoom of these three places along the axis. As you can see that it's really changing the orientation which correspond to the activation pattern that we see. So to conclude, we showed just that the shape analysis could be interesting and provide some insights, and we can link it to cortical folding and function, and maybe more systematic study could be carried out to further the study, for example, the link between the cortical folding and the psychoarchitecture. Thank you for your patience.